Hey guys, so we are still keeping with the histology theme and we are going to do the urinary system in this video. So with the urinary system, we are going to start where the blood gets filtered and starts the whole process of creating that urine as filtrate. We are going to start in the kidneys. So on the kidney, I've got it zoomed way out. We are on scanning power, and I have this just because I like to show all the different parts of the kidney in one place. So we've got the capsule on the outside, like candy covered shell, like an M&M is the capsule out here. Then this kind of lighter area from like here to here. This kind of section is the cortex, and this kind of darker stuff would be the medullary area, medulla, on the kidney. On the zoom out, we can see all these things that look like little balloons here. These are going to be corpuscles. And then we can see lots of little open like tubes and ducts running all throughout the kidney. So all different things going on in this slide. When we zoom in on the next picture, we can see that there is a zoom in on a corpuscle. So this whole entire thing, this is all a corpuscle. Inside the corpuscle is made of two parts, two things in a corpuscle. First thing is what's inside, and it is this guy. It is the glomerulus. The glomerulus. The glomerulus are where we're going to have the blood flow feed in. We're going to have an afferent arteriole come into this primary capillary bed. It's the glomerulus, and it's going to undergo filtration. It's totally non-selective, and that filtration is going to form. We're going to form a whole bunch of filtrate. This liquid that's going to come out of the blood is going to be filtrate. So we're basically pushing anything that can fit through the holes before and in between the little podocytes, in between those slits, out. Anything small, water, lots of different small things will slip out, like glucose and sodium and all that stuff comes out in the liquid as filtrate, and that gets captured in the second part of our corpuscle. So it gets captured here. So this is the Bowman's capsule. So together we have the glomerulus, number one, and the Bowman's capsule is number two, and together they form a corpuscle. Both of these structures, the glomerulus, which is our primary capillary bed, where we are doing non-selective filtration, and the Bowman's capsule that kind of wraps around the glomerulus and captures the fluid in the Bowman's space, are going to be simple squamous epithelia. Simple squamous, because they are simple squamous epi. Because they are simple squamous, they're very thin, and we said simple squamous was really awesome for filtration and diffusion of gases. So filtration is where it's at in the kidneys. That's what we're doing. We are filtering the blood and pushing everything out that we don't want, and it's going to go out into this Bowman space as filtrate, and then it's going to leave that portion. It's going to leave the corpuscle, and it's going to leave that portion, and it's going to go into the convoluted tubules. The first convoluted tubule that we see is the proximal convoluted tubule because it's closest. The furthest convoluted tubule is going to be distal. So PCT, proximal convoluted tubule, DCT, distal convoluted tubule. The PCT, this guy right here, is our first stop. So we said we collected all of this filtrate. All this filtrate is being formed, all this liquid. It's like 125 mils a minute. It's ridiculous. 180 liters, I think, a day. Huge amount of water, huge amount of solution that we are creating that is filtrate, and it is going through into this space. So this is going to be our PCT right here. And so on our nephron, just to describe, here's our little corpuscle. I got some, you all know my doodles are not awesome, but they kind of give you an idea. This is PCT, proximal convoluted tubule. We're going to go down, we're going to come back up, go down, going to come back up. This is the loop of Henle. And then over here we have the distal convoluted tubule, and then we're going to go into a collecting duct. Distal convoluted tubule, collecting duct, DCT, and that whole thing, Bowman's capsule, this whole thing is a nephron, and these are the different parts that we can see on the slide. Collecting duct, collecting duct. All of this is the nephron. The nephron is the functional unit of the kidney, and we can see nephrons all in this kidney. So the first part of it, again, was the Bowman's capsule, and the glomerulus. Together, they make the corpuscle. They are going to have non-selective filtration. Fluid comes out, and it goes into the first stop, which is the PCT. Here's our PCT on our slide. Here's our PCT. What do we need to know about the PCT as far as lab? The lab portion, we need to know what type of epi we have in the PCT, in those convoluted tubules. PCT and DCT are both going to have the same type of epi, and that is going to be simple, but instead of squamous, we are now a bit bigger. We're chunkier. We're as tall as we are wide. These are simple cuboidals. So roughly about as wide as they are tall. About as wide as they are tall. These cuboidal ones. PCT edges look a little bit fuzzy. They kind of have this like cilia brush border look to them on the PCT versus the DCT where they've got a little bit cleaner edges. But those are going to be our simple cuboidal cells that line the PCT and the DCT. On our Bowman's capsule and glomerulus, those are going to be simple squamous epithelial, real thin. And you can make out how thin they are. They are super thin, little pancake ones that make the capsule around everything. And they also are going to line this capillary bed that's all kind of smushed up here called the glomerulus. Next up on the kidneys are DCT, and we can also make out, so here's another, these are DCT, all of these are DCT. And remember we said convoluted tubules are all going to have simple, because these are absolutely just one layer thick, simple cuboidals. And we have lots of different things over on this side. So we've got a glomerulus over here. We got a Bowman's capsule over here. We got PCT coming out. And then it looks like we may even eventually do a cross section and go down. Some of these are going to have really long areas. This might be one, but I believe this might be part of the PCT. But in some areas, let me back it up and see if I can find another one. Ooh, here's a better one. This guy right here, collecting duct. So the collecting ducts are going to empty out several nephrons, and the collecting duct's going to go down. There's going to be lots of different collecting ducts, and they're going to go out of a place called a renal papilla, and all the fluid that comes out of there is no longer called filtrate. It's going to be called urine. But these real long skinny ones, these are collecting ducts in this image here. So those are all of our different terms as far as the kidney and its histology. Next up is the bladder. We have created all this filtrate, and we're going to store it. So where do we store it? We store it in the bladder. That is our storage center, and it can hold quite a decent amount of fluid in there. When we have just gone to the bathroom, our bladder is relaxed, and you can see we've got lots of wrinkles going on here. It's nice and relaxed because we just went to the bathroom. But if we'd been holding it all day and we had, I don't know, a big beverage for breakfast and a big beverage snacking throughout the day, and we had a big lunch and lots of water and fluids in our meal, we might have a very distended bladder if we did not get a chance to go to the bathroom yet. So look how flat this is. This is a full bladder, really distended compared to our relaxed bladder that got to go and relieve themselves. Because our bladder is able to stretch like that, the epithelium can stretch like that. So the stretchy epithelium, what we talked about in AP1, is transitional epithelium. So here, when we zoom in on that bladder, all these cells, all of this is transitional epi. So all this stuff, this is all all these guys, these are all transitional epi. They are super stretchy. They are a type of epi, so they're going to have an apical end at the top. They're going to have a basilar end. Where are my basilar cells? Basilar end at the bottom. They're going to sit on top of the lamina propria down here. And this is the lumen where the urine is stored in there. So that is the bladder. Know the type of, recognize the bladder, and also know the type of epi that is found in the bladder, transitional epi. Heading and connecting from your two kidneys down into the singular storage unit, which is the bladder, is going to be our ureters. And so we got a side of the ureters. This is the lumen, the space here that's going to transport that urine down from both kidneys to ureters, meets at the bladder. So this is also going to be stretchy. It's going to be stretchy. So we have transitional epi that's going to line this. So here's our lining. This would all be urine in here. And these cells right here that are like all this guy right here. This is all the transitional epi. So this is going to be our mucosal area. We got our lamina propria again, all this light pink stuff going from here to here. And then we are going to have smooth muscle, just like we talked about in the GI tract, where we've got peristalsis moving from point A to point B. So we have a smooth muscular layer. So even if we were standing on our heads, we could still move that urine from our kidneys to our bladder, no matter what. So here is the muscle layer on the ureter. And then our last layer is that connective on the outside, and that is called adventitia. So here's the very outside. Nope, just that side is the only one that we can see the edge of it. But this is going to be our adventitia. Probably a little more over here, a little bit over here, a little bit over here would be the adventitia, the connective area that keeps the ureters in place where they need to go. So those are the last bits of the urinary histology. I hope this helps you, and I hope you have a wonderful rest of your day.